Today we're going to look at DNA fingerprinting. Now, this is different than your fingerprints on your hand, but it does make an identifier. So for example, we are going to do a gel electrophoresis. So this is agar, which is like a fancy science jello, and it has these little wells in there. And so what we do is we put a known marker or a dye, it's also known as a ladder, in this first column. And then we put our other suspects or other DNA samples in these wells. DNA is negatively charged, so it moves towards the positive end. Larger molecules move slower, so these are longer molecules, and these are much shorter molecules. We'll run electricity through this, and it separates out. Okay. So what we're going to do is we notice that this one and this one line up exactly. So these two are what we call a DNA fingerprint or a match. The purpose of doing this is for a number of reasons. Okay. For paternity or maternity, related genes such as cancer, forensics, as well as breeding purposes. Okay. So for example, a DNA sample is taken and we use hair tags, we use saliva, blood, or semen. We also use skin as well because there's DNA in your skin. Number two, small samples are copied by using a PCR. It's also known as a polymerase chain reaction. And we've seen this word before, polymerase. Next, DNA is cut into specific locations. For example, CCGG is also known as a restriction enzyme. And we cut it between those two. The cut DNA is then placed into the well. DNA is negatively charged and moves towards the positive pole. Lighter segments move farther from the well, and we compare it to known samples.